Hey guys, welcome back to Computational Biology. In this lesson, we're going to be going over seek objects. And so what seek objects are it's kind of like taking strings or, collections of letters, like a string would be like a word or a phrase or things like that, and turning it into a biology relevant string. So we can perform functions on them or manipulations to them that are unique to a seek object and not to a string, like concatenating or adding sequences together and things like that. This is part of the Biopython package in Python. So if you don't have it installed, you should get it installed to be able to work with it. So let's just jump right in. Okay, so like I said, the most important difference between a seek object and a standard Python string is they have getting text messages, is that they have different methods associated with them, and that's part of the point, I guess, of making them a seek object. Although the seek object supports many of the same methods as a plain string, it has things like translate methods to translate the FASTA file or the sequence into amino acids, etc. So let's start here by opening. Make sure you're in your My Files. I'm gonna open this book and I'm going to call this. Let's call it just seek objects. Okay, so the first thing I'm going to do from bio.seek import capital, yes, seek. I'm going to run that. Actually, before we do this, no, I'm not going to cancel. So I'm going to go File, New, where's launcher? New launcher. This might take a minute to load up. So Praxis has moved their servers to a different computational center, so they're a little bit slower to wake up because they have to boot up a new machine. It was explained to me every time. So you have to be a little patient with it. But this allows them to scale up. So I believe it's pip install biopython. I hope that works. Okay. So this might take a second to install. Successfully built. All right, so now we'll go back here and let's try running this again. There we go, now it works. Okay, so let's keep going. So that just loads up the package. So my seek equals seek. So we're making a sequence object as opposed to a string, GATCG. We'll run this. Now for index, letter, and enumerate my seek, what we're going to do is we're going to give numbers to each letter or nucleotide in the sequence. So print %I %S. Oh, and these, yep, like that in quotations, and index letter. Okay, so let's run this. And as you can see, now we've given a number, and remember Python's weird, so the first thing in the sequence is always 0, so we could count starting at 0. We can also do stuff like print len my seek. If we do this, oh, as a reminder, as I said in my intro video, you are expected to provide your own annotations for stuff like this. So while you're doing this, you can say we can also print the length of each sequence. So it's your responsibility to annotate for this second part of the course. Okay. Print k. So just like with strings, though, we can also access the different positions the same way with seek objects. So we can do print my seek 0, and so that gives you G because that's the very first position here, right? And we could do print my seek, we pick a number up to 4, so that'd be the last object. So if we did though print my seek 5, we get the error, right? Because we only go 0 to 4. So let's change this to 2, just so we don't have an error in our thing. So you get the T, etc. We can also do something like this. We can do a .count function. So say we'll make a new seek object called AAAA, and we will do .count. So we're telling it to count the occurrences of AA. So how many times does AA happen together? And so you would guess that this would be 2, right? Because it counts 1, 2 for the first, and 1, 2 is the second. It doesn't count overlapping, right? So it doesn't go 1, 2, through, and then the middle 2, 3, 4, 5, 6. So it doesn't do that. So we get 2, right? Where else to go now? We could also do my seek equals, and we'll put a sequence in here. Jake, you can just do whatever nucleotide. Make sure it's just all A's, G's, T's, and C's. And then we'll run that. We can do the length of my seek. That should be capitalized. There we go. Length of my seek, so that's 32 letters there. We can also count the letters in a specific letter, so my seek .count, and then we can give it what we want it to count, like we did up above. So we can say, okay, now count all the G's. And you see we have 9 G's in our stuff, in our sequence. We can also do, so why would you want to do that? Well, one thing that is important for doing things like designing primers and things of that nature is measuring the GC content. So we could do 100 times my seek count G and my seek count C divided by the length of my seek. So this should give us, get rid of that there, 46.875% of our sequence is G's and C's, which is wonderful. So the other thing, so things like that are handy, right? But also a lot of the common stuff, like calculating GC content, is already built into Biopython. So what we'll do for that, so let's do from bio.seek utils. Let's move that out of the way. Hold on, let's see. I'll make this a bit bigger for you guys. There you go. Sorry, that was not the largest. Put it right here so my picture's on there. Okay, bio.seekutils, import GC fraction. So we've imported the GC function. So we'll do my seek equals and then just make up a sequence. Okay, so I think, oops, there should be a Y in there. I think that's the same sequence I typed before. So we should get GC fraction of my seek should be the same. Yep. So essentially this has been times by 100 give you percent. This is just the decimal. Okay. What else do we have to go over here? So we can also slice sequences into multiple parts. So let's do, we'll just use that. Okay, so what we can also do is we can do slices with a start and a stop and a stride, it's called. So for example, let's do my seek 0. So we're going to start at 0, or the very first base pair, and then we're going to say cut out every third nucleotide, right? So if we run this, you should see that we start with G, perfect, and then we skip 2, and then the second letter is C, and then we skip 2, and the third letter is T, we skip 2, and then G. So that is, so our stride is pick every third. So skip 1, 2, and then include the third. Let's do another one. So my seek, if we did the same thing, but we'd start at position 1 instead of position 0, then we should get different letters. So we started at position 1 here, skip 2 to get a G, skip 2 to get a G, etc. And we'll just do one more just for the heck of it. So this is important for, remember, because we're reading DNA in codons, right? And so we read DNA or RNA in letters, 3 packs of letters. And so a GAT is a codon, a CGA is a codon, a TGG is a codon, etc. But depending on where we start, our nucleotide or our amino acid sequence is way different. So if we start at the second position, then we have ATC, GAT, GGG, which is way different. And so it's like a frame shift mutation, right? So if you've taken genetics. So yeah, so we can choose where we start, our start position. So just like with strings, we can use negatives to start from the other end. So if we do my seek ::(-1), it'll just print the entire thing backwards. So here it is, just backwards. Let's see. Oh, if you want, so say you have a seek object and you want to turn it back into a string. How do we do that? Pretty easy. str and then mySeek, and that will turn it back into its string. You can save it as a variable or save it to file or whatever you want to do. So you put the arrow and label a variable. What else can we do with seek files? That's kind of cool. Let's see. Let's do FASTA format string equals name. And %symbol mySeek. Print FASTA format string. Okay, so what we have done is you can have like a verb placeholder when using the Python string formatting. So in this case, we gave it this name to denote what our FASTA string is. If you want to have like labels, let's see. So this is like FASTA format, right? Where you always start with this greater than symbol. What else can we do? There's so many things. Let's do one more and we'll call it for this video. What's important? Concatenating. So concatenating is adding, right? So you're adding two scripts together. So let's do seek1 equals seek, not minus, equals seek ACGT will be our first sequence. Seek2 equals seek, and you can pick whatever letters for yours, it doesn't matter. And then so I'm gonna run these quick. So now we have a seek1 and a seek2. If we just do seek1 plus seek2, it'll intuitively add them together, right? And if we do it the other way, seek2 plus seek1, it'll add it together in the other orientation, where it starts with AA for the beginning here. So yeah, I think we'll stop there. We'll go through more in the next video here. There's a lot, and I'm trying to kind of like weed out as I go. Like, what do you really need to know? So these are some of the most common like ways for manipulating sequences. And the seek object is nice because it makes it really easy to do the common things that you would do: add two sequences together, or splice one, or pick out where your the codon that you want, or the base pairs that you want, and things like that. So we'll keep going with seek objects in the next one. Thanks for watching. If you're watching on YouTube, please hit like, please hit subscribe. It really does help me out. And I'll catch you in the next video.